let's start our webinar. Bismillahirrahmanirrahim. The Honorable Vice Rector of Research, Innovation, and Partnership Universitas Hasanuddin, the Honorable Dean of Agriculture Faculty Universitas Hasanuddin, the Honorable Speaker and Moderator, and
recording in progress.
The Honorable guests and participants. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Ladies and gentlemen. My name is Israeni Yulanda Iskandar. It's a wonderful and precious chance for me to be your Master of Ceremony on this occasion on Tuesday, the 5th of April 2022, in our webinar, Healthy Soils with High Diversity Are the Way Forward for a Sustainable Agriculture, How Agroecological Practices and Microbial Biotechnologies with Beneficial Microorganisms Contribute to this Goal, organized by the Faculty of Agriculture, Universitas Hasanuddin, to the World-Class University Program. First of all, let's say thanks to Allah Subhanahu wa Ta'ala, who has been given us guidance, happiness, healthy, and mercy, so we can attend and participate in this event virtually. At this beautiful moment, I would like to welcome Professor Dr. Didier Lesser, who will be the speaker for our event today. Good morning, Professor.
Good morning.
It's a pleasure for Universitas Hasanuddin to have you as a speaker today. We also would like to welcome all participants today for their presence. Thank you, ladies and gentlemen. Then we have several agendas for this special event, so please allow me to read several for our agendas today. The first is the opening. The second is remarks. The third is the main agenda, which is the presentation about healthy soils with high diversity are the way forward for a sustainable agriculture, how agroecological practices and microbial biotechnologies with beneficial microorganisms contribute to this goal from our speaker and discussions. The sixth is photo session, and the seventh is closing. All right, ladies and gentlemen, the first step is on the following agenda is remarks. The first remark is the welcome remark that will be delivered by the Dean of Agriculture Faculty. I immediately invite Professor Dr. Insinyur Solenke, MSc, to give his remark. Please, Professor.
Thank you. Ladies and gentlemen, good morning.
Good morning, Professor.
The Honorable, our distinguished speaker today, Dr. Didier Lesser. I'm sorry if I pronounce it wrong.
Very good. Well done.
Thank you, sir. Thank you, sir. And all other attendees, thank you all for being with us here today. I'm pleased to welcome all of you to this Top Scientist webinar series hosted by Faculty of Agriculture, Hasanuddin University. And today we are very fortunate to have Dr. Didier Lesser as our speaker today. Dr. Lesser is a senior soil microbiologist from at CIRAD, from Montpellier, France. In addition to his position at CIRAD, Dr. Lesser also served as associate senior researcher at the Alliance for Biodiversity International, adjunct associate professor at Deakin University in Melbourne, Australia, and also as visiting professor at Chinese Academy of Tropical Agricultural Sciences. We are very fortunate to have you talk to us today, sir. In this webinar series, Professor Lesser will talk about healthy soils with high diversity as a way to achieve sustainable agriculture. This webinar series is designed as a venue for scientists from academia, from research institute, to share their knowledge, their expertise with us at UNHAS and researchers at other institutions in Indonesia. I see that the attendees now is more than 160. This is from all over Indonesia. Welcome you all to this webinar series, and I hope that we will have fruitful discussion during this event. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor. All right, the next remark is an opening remark. It will be delivered by Vice Rector of Research, Innovation and Partnership, Universitas Hasanuddin, and continue for the opening of this webinar. However, this occasion will be delivered by video because today, the Vice Rector of Research, Universitas Hasanuddin, has another schedule, so we apologize for that. Please, the IT, could you please play the video of our Vice Rector, Professor Dr. Muhammad Nasrul Masih, PhD, SPMK. Pak Tami.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, the Honorable invited speaker, the Honorable Dean of Faculty of Agriculture, and distinguished moderator and participants. Ladies and gentlemen, firstly, let's us praise and send great gratitude to Allah, the Almighty God, for His blessing, mercy, and guidance upon all of us, so we can attend this webinar in a healthy condition. On behalf of the university, it is an honor for me to open and welcome you to the Top Scientist Webinar Series, Faculty of Agriculture, which is conducted by the world-class university program Universitas Hasanuddin. Special thanks to Dr. Didier Lesieurs for your presence and agreement to share a valuable topic with us. Ladies and gentlemen, the theme of this webinar is entitled Health Soil with High Diversity Are the Way Forward for the Sustainable Agriculture. How agroecological practice and microbial biotechnologies with beneficial microorganisms contribute to this goal. Healthy soil is a crucial thing that allows plants to grow to their maximum productivity. It contains a high diversity of bacteria, fungus, and protozoa, and other tiny creatures. Those organisms play an important role in growing healthy plants. Soil bacteria take a role to produce natural antibiotics that may help plants to be resistant to this disease. This condition is the nearest way to achieve the sustainable agriculture goal. In this webinar, our speaker will focus on discussing how agroecologists and microbial biotechnologies take a role in utilizing beneficial microorganisms and finally achieve the goal. However, agroecologists and microbial biotechnologies contribute a big part to sustainable agriculture. We ensure that all participants will get more complete information in this webinar. Ladies and gentlemen, this webinar today is provided by the world-class university program of the Ministry of Education, Culture, Research, and Technology. Through this program, we are being supported to gain more collaboration with international colleagues in scientific activities. Therefore, Dr. Didier Leisure that can be profitable for both of us. And with that, I would like to conclude my remark and officially open this webinar. Bismillahirrahmanirrahim. With the blessing of Allah, God Almighty, I officially open this webinar. Wabillahi taufiq wa hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi. Thank you very much.
All right, let's keep on the next agenda. The main agenda of today's webinar about healthy soils with high diversity are the way forward for a sustainable agriculture, how agroecological practices and biotechnologies with the beneficial microorganisms contribute to this goal. From our speaker, Professor Dr. Didier Leisure, he is a senior soil microbiologist at CIRAD, UMR EcoSols, Montpellier, France, and he is associate senior researcher at the Alliance of Biodiversity International and CIAT Hanoi, Vietnam. He is also adjunct associate professor at Deakin University, Melbourne, Australia. And he is also visiting professor at the Chinese Academy of Tropical Agriculture Sciences, Rubber Research Institute, Hainan, China. And the next agenda will be conducted by our moderator, Muhammad Junaid SP MP PhD, the lecturer of Agriculture Faculty, Universitas Hasanuddin. Dr. Muhammad Junaid, time is yours.
Okay, thank you very much, Isra. This is nice. So first of all, I would like to thanks to all the participants, the honorable speakers, Dean of Faculty of Agriculture, Vice Rector for Collaborative, and all participants in this room platform. And actually, I would like to tell behind this before we doing the webinar that all participants in this here, Dr. Didier, they are from our students, from undergraduate and postgraduate. They are really interested in doing, in joining in our seminar. That's because of their very connecting with the backgrounds and what do you want to talk today. So this is our background story about. Then we want to say in your presentation, I would like to say that this is a very interesting talk. That's because of healthy soil, sustainable agriculture, and practicing in agriculture. We know now in our Indonesia, so most of our farmers doing that unsustainability, and so many consequences of that. And hope with your lecturing today, we will get a new insight for our students and how the agroecological practicing and soil microbiomes or biotechnology of the microbiomes can contribute to a healthy soil, healthy plant, and healthy environment. That's why sustainable agriculture is very important. Well, I don't want to be like a speaker. Good morning, Dr. Didier. How are you?
Good morning, Jung. I'm very good, thank you.
Okay, this is time for you now. Go ahead. Yes.
Okay, so I can start by sharing my screen. Let's hope it's going to work. Can you see my screen?
All right, good.
Okay.
Yeah.
So first of all, okay, let me start the time. First of all, I would like to thank the University Hasanuddin University for giving me the opportunity to make this webinar. And it's a great pleasure to see that so many people are connected. So I hope that their expectation will be fit by the content of my presentation. At the same time, if you have questions, don't hesitate to interrupt me, and I will be happy to answer your questions. So I tried to gather some information about agroecology, about soil health and everything. So this is the outline. So I will start by some definitions. After that, I will explain what do we call soil health and what are the main factors we have to take into consideration when we think about soil health. I will give you some examples of two important nutrient cycles involving soil organisms, the nitrogen cycle and carbon cycle. And I will make a brief introduction about Asia Pacific CMBP network, because your university is part of this network since almost the beginning. So it's quite important. Maybe you are not all aware about that. And I will explain you what we are doing, and not just matter of talking, but also I will show you some examples of collaborative activities we have been running in Vietnam with national partners, and it will give you some idea about what it will be possible to do with partners in Indonesia. So let's start by some very basic definitions. I'm sure you are all very much aware, but for me, by showing this definition about agronomy and listing the scopes of the agronomy is to show you the importance of soils. Now, I mean, 5 years ago, people were not very paying attention to soils. Things are changing, fortunately. I think it was time. I hope it's not too late. But now you can see that FAO, for instance, the United Agency organize every year a soil day on December 5th, I think so. So they are organizing in every country's big events just to raise the issue about why is it so important to take care of the soils. So you can see, because soils is key for agronomy, so it means for agriculture, and there are several ways how to improve agronomy. So in the title, I put the terms of agroecology. Agroecology could be called sometime climate smart agriculture. They are almost the same. I don't know if you are very familiar with this kind of vocabulary, but in fact, agroecology is a way to enhance soil diversity. It's a way to reduce the utilization of chemical inputs, but also in maintaining good yields. So sometimes the balance is not so easy to find, but we have success stories. And I mean, if I take the example of Vietnam, I mean, farmers have been using too much chemical inputs with huge negative consequences on their health, on the soil health, and at the end of the day, the system becomes totally unsustainable. So we have to change. Agroecology is definitely the way forward if we want to be successful, but it takes time to convince farmers to adopt this kind of practices. So soil health, very important. I mean, in English, it's stronger than in French. When you say in French, people, sometimes they are confused between what does it mean? Is it about the plants? Is it about the soils? I think I like this definition I put on my slide. Soil health is defined as the continued capacity of soils to function as a vital living ecosystems that sustains plants, animals, and humans. It's very important. And why is it so important? Because it plays a role in the regulation of water, indeed, like a filter, catching water and everything. Sustain plant and animal lives, because it provides a lot of nutrients that all these organisms needs. It can be also a kind of buffering system for when there is pollution up to a certain limit, indeed. And of course, because all the main cycling nutrients happens in the soils, carbon, nitrogen, phosphorus, and so on. They are stored in the soils, they are transformed in the soils, and this is where the plants go to get it. And of course, in terms of support, the plants cannot grow except in hydroponic system, but it's very limited. Without soil, it's very tricky. So that's why soil health is so important. And it has to be the center of whatever we try to do if we really want to sustain the agriculture. So just an illustration about what I was saying a few minutes ago. Soils in ecosystem, you see in terms of carbon sequestration, climate regulation, nutrient cycling, habitat for organisms, food regulation, and so on and so on. It's very key. Yeah, I mean, for the last 2.5 years, we have all been struggling with this COVID-19. A lot of theories about what happened, and I'm not going to talk about any hypothesis, but on this figure, you can see the starting point is soil degradation and desertification. So which means land misuse and soil mismanagement. A lot of consequences of this with soil degradation, food insecurity, problem with human health. And you see now, it's where you start getting pandemics. So of course, I cannot say that's why we got this problem with COVID-19, because I don't know about the starting point, but just to show you that some people believe that because of global warming in the ice, maybe some viruses are there, they have been released by the melting of the ice, and also it may be all consequences of some pandemic. I don't know, but just to show you that up to where soil degradation can go and the consequences for humans. I'm going to try to show you a video, but I don't know with, okay. This video is nice. I used to show it to the students. Let me check if I can see it. I cannot see it on my screen. I don't know why. Yeah, oh, okay, here we are. I need to be sure that you will be able to hear the sound. Can you hear it?
Yes.
All right. Okay.
No, we can hear the voice. Yes.
You can just have a voice. Yeah, that's why I don't know how to make. Okay, let me check. Because I used to, I'm working with Teams when I give. Just a minute. Huh? Oh, okay, here it is. Okay, it's a bit different with Skype. Is it working now?
Yes.
Yes.
Okay,
Yes.
let me put full screen, and we can go ahead.
That distant and small spot in the cosmos is the habitat of approximately more than 2 million species and the product of billions of years of evolution. Most of this biodiversity is in the soil beneath our feet, but only 1% of soil species have been identified so far. The soil biota constitutes the greatest concentration of biomas of any part of the planet and is composed of an immeasurable amount of mega, macro, meso, and microorganisms. Each of these wonderful creatures plays a key role in the food chain that maintains the fragile balance of life in natural and agroecosystems. If these complex interactions become interrupted, it can cause irreversible impacts for life on Earth, including humans. So, why do we need soil biodiversity? Soils hold many unexpected secrets and perform unseen functions. The discovery of antibiotics has had a major impact on increasing human life expectancy. Soils hold many other potential medicinal functions and possible cures. The natural association between plant roots and microscopic fungi promotes better plant nutrition and growth, tolerance to soil pathogens, and adverse climatic conditions. Through their natural function and metabolism, soil microorganisms are capable of breaking down and denaturing certain toxic compounds and contaminants resulting from many human activities. Part of CO2 emissions into the atmosphere, derived from industry and agriculture, can be absorbed by plants and stored in soils thanks to microbial decomposition that may allow soil carbon retention for long periods of time. This invaluable service provided by soil organisms is key in climate change mitigation. On the other hand, deforestation, monocultures, and the overuse and misuse of agrochemical inputs degrade and reduce soil's health, diminish resistance to pests and pathogens, and cause biodiversity loss that jeopardize the delicate balance that took billions of years to evolve and specialize. We could not only lose a long natural history of speciation, specialization, evolution, and adaptation, but also soil's capacity to perform essential ecosystem functions. How can we restore, manage, and conserve something that we do not see directly, and of which we do not have full knowledge? It is our collective responsibility to raise awareness on the importance of soil biodiversity, promote technological innovation to preserve and enhance soil biodiversity, including ecosystem restoration, recognize soil biodiversity as a key provider of ecosystem services, and as one of the main nature-based solutions to face all the current global challenges. Invest in gathering better knowledge about the status of soil biodiversity and functions, including by region and land cover type. And develop policies based on scientific evidence to mainstream sustainable soil management and conservation of rich biodiversity soils across landscapes. Everything that we eat and drink passes through the soil biota over and over again. Therefore, we need healthy soils for healthy food, healthy environments, healthy people, and a healthy life. It starts with you. Give a voice to the sound of a living soil. Keep soil alive. Protect soil biodiversity.
Okay, so I can go back to my screen. So you can see the message is pretty straightforward. And that's why I like using this video, because I think that they go through many issues I'm going to talk about right now, just as an introduction. So as they said, soil biodiversity is very much important because soils contain so many living organisms. Yes, just to give you an example, 1 billion bacteria cells in just 1 gram of soils, several thousand of different taxa, 200 meters of fungal hyphae. So it shows you a little bit, and they are so diverse. I mean, you can get bacteria, you can get archaea, amoeba, protozoa, also herbivore nematodes. So it's quite diverse. And this diversity is very much important. So we can also try to define what is soil biodiversity. And I mean, it's a variety of all the living organisms living in the below ground soils. And they form some community, they are organized in structure, and they contribute somehow, they all contribute to the main, what we call ecosystem services. And of course, soils is one of the main global reservoirs of this biodiversity, because we consider that more than 40% of the living organisms are in the terrestrial ecosystems. So we had to define this soil biodiversity, and experts decided to use the size of the living organisms. So that's why we have from the smallest microbes from 20 nanometer to 10 micrometer, and the microbes on microfauna, they are 10 micrometer to 0.1 millimeter. After that, we move to mesofauna, macrofauna, and what we call megafauna. And this classification is still used. And when people talk about microbes, it gives you an idea about they are the smallest organisms, and so on and so on. So of course, soil is quite complex. We may think that it looks like something very homogeneous, but it's not at all. It's very heterogeneous. It is because of the size of the particles we have in the soils. And according to this size, of course, the distribution of the living organisms will be different. And this can be strongly modified by agricultural practices. It may be tillage, it may be also utilization of chemical input. So it seems quite solid and stable, but it's very fragile. And somehow the sustainable system can become totally unsustainable because of this. So that's why we have to be very careful. And I was mentioning the importance of soil structure. So you can see, because in fact you have a lot of aggregates. I will give you more details about what we call aggregates. But you know, the aggregates are part of the soils with quartz particles, with clay particles. So it means that for living organisms, they have so many niches where they can stay. And when they have got favorable niches, they can grow. Sorry. They can grow, and they can start their actions. They can start, for instance, storing carbon and whatever. So the system becomes quite sustainable. But this system can be totally broken down if you do tillage, because of course this structure disappears and now you start from scratch again. So this is very much important. So as I explained you a few minutes ago, all these organisms are classified according to their size. So I'm going to show you examples of each category, starting from the smallest microfauna, and we will go gradually to the biggest. Why is it so important to talk about soil biodiversity, and how do these organisms use to interact with each other? This is something very much important that we need to keep in mind, is the food web. Food web is, what does it mean, soil food web? It means that they all depend to each other. So bacteria and fungi are part of what we call soil organic matter. They are very important because they play a direct role. I'm going to show you which kind of roles they used to do. But at the same time, they are also important because they are food. They are food for protozoa, for nematodes who used to eat bacteria, or nematodes who used to eat fungi. And this nematodes or protozoa, they have their own roles. They are also food. They are food for predator nematodes. And all these predatory nematodes can be eaten by microarthropods. And you have also arthropods eating fungi. And all of them, they used to interact. So it means you have three big groups, what we call ecosystem engineers here, hence termites, earthworms. We have what we call litter transformers. So organisms were able to transform the organic matter in something else. I'm going to give you the names and some explanations. And you have these kind of interactions. So it's very important because at the end of the day, the ecosystem engineers, if they cannot get food, or even the litter transformers, if they cannot get food, the system is not going to work, and the ecosystem will get stuck. So that's why it needs to be very balancing on this. What we call soil food web is very much important. We have to pay attention because we cannot get one category working alone. The three categories need to work together. So let's talk about the soil bacteria. So you can see from these photos that they are quite diverse. And we are very abundant, as I said, because we have several billions of bacteria in 1 gram of soils. And it was very tricky at the beginning for people who started investigating bacteria, how to define them, how to classify them. And but now with molecular tools, we know we have some ideas about the classification of this bacteria. And we have discovered that they are very important because they may be decomposers, they can be mutualists, they can be symbiotic pathogens, and of course, they play a key role in nutrient cycles. And I will show you later. Soil fungi. Yeah, they are, first of all, they are very important for structuring the soils because physically they can bind the soil particles, and they also strongly contribute to the formation of aggregates. Because they play a role in the porosity of the soils, they are also a key factors for water dynamics. And I was mentioning about organic matter. They are very much important in terms of decomposing the organic matter to release nutrients that bacteria and plants will be able to use. And in some cases, they're also very much important, like bacteria, as biocontrol agents. And as for the bacteria, they can be mutualists, they can be symbiotes, decomposers, and so on. Now, I mean, there is nothing new for you about bacteria and fungi, but now viruses. For many years, they were totally neglected. No one believed that we had viruses in the soils. But as you can see on this figure, viruses are very much important because they have a lot of interaction with other soil organisms. So in some cases, this kind of interaction can be positive, such as here. You can see that they have the capacity to kill pathogenic bacteria or to kill pathogenic fungi or pathogenic, other soil-borne pests and diseases affecting the plants. But sometimes also they can be negative for the plants because we have some pathogenic viruses and they affect directly the plants. So basically, it means that even if this packets of DNA of RNA, they don't have a direct role, they shape the microbial of microbial communities, sometimes in the positive way, sometimes in the negative way. And indeed, as I showed you previously, they are very much important because they have a big influence on the soil food webs. And that's why we have to take them into consideration. Another group that people neglected for a while, it's the protist. Oh, what is the protist? We don't really know, but they are amoeba, ciliates, and flagellates. They are very important because they have a big role in terms of nutrient cycling, direct, because they release nutrients when they eat bacteria, but also indirect, because they influence the soil structure. And according to the soil structure, the availability of nutrients can be different. And somehow, we have new recent publications showing that, but protists also can have a biological regulation role by shaping the composition of the bacterial communities, as I said, for the viruses. So you have an example of kind of protist. And for your information, we are about, I think I share this position through the network. We are seeking for someone who has knowledge and protists and nematodes, so it will be my next slides. And if we are successful with this selection of this candidate, we will have a scientist based in Hanoi working on this. And of course, through CMBP, that will be an expertise that we will be able to share with our members. So just keep in mind that we will get someone who will be able to help us working on this. So soil nematodes. For many years, we consider soil nematodes as negatives, because we had in mind that all the nematodes were bad for the plants, which is not really true. And more than that, we discovered that soil nematodes can be very good soil health indicators, because their composition can quickly change from one condition to another one. And we have so diverse groups of nematodes. We have bacterivores, fungivores, herbivores, predators, omnivores. And so that's why we are very much important. And that's why you can see now people, they are trying to use nematodes communities as a very good soil health indicators, because they are very sensitive to the environment, such as carbon transformation, nutrient cycling, soil structure, and so on. So just a quick, if you remember, for what we consider as very important soil organisms: bacteria, fungi, viruses, protists, nematodes. Okay, so how does it work? So this is the example of a carbon cycles, or you can see how it works between the earth and oceans. And today I'm going to talk just about what is happening here. And because this is where the microorganisms are very much important in terms of microbial respiration, in terms of decomposition of the organic matter, with indirect effect and photosynthetic bacteria and so on. So let's talk about soil respiration. What does it mean? This is the situation. You have your plant, you have your soils, and of course, soils, the plant does photosynthesis. So she's able to get CO2 from the atmosphere, and in exchange, she releases oxygen. But in the soils, we have roots. So there is a turnover of the roots with release of organic matter. We have some letters coming from the leaves. And of course, this organic matter is going to be transformed by soil organisms. And when it happens, that's what we call soil respiration, because this organic matter becomes source of carbon, and with oxygen, these organisms can transform it, and they produce energy, water, and they release some CO2 in the atmosphere. So you can see that this involves a lot of organisms, symbiotic microorganisms such as mycorrhiza, and you have also some rhizobia if this is the legumes. You have also the impact of decomposers and so on. Okay, so next step is organic matter. This is the starting point. So the organic matter comes from many sources. It can be dead forms of organic materials, living parts of plants. I mentioned the litters, the turnover of the roots. Can be also living microbes and soil animals, but they are going to be consumed by predators. And it's quite diverse. And so who is in charge to decompose this organic matter? They are mainly what we call saprophytic fungi. So right now the plants from this organic matter, most of this organic matter cannot be used by the plants. It needs to be transformed in smaller molecules, such as organic acids. So basically, it means that from this organic matter, the saprophytic fungi are going to decompose this organic matter and make things available. So at that time, bacteria are going to play their role. They will transform them, and at the end of the chain, the plants will get the nutrients she needs to grow up. And of course, we will observe this emission of CO2. Some bacteria, just for your records, some bacteria are also capable to decompose the organic matter. They are not. Most of the decomposition is done by fungi. So this is the scheme of what is happening with soil organic matter decomposition. We start from organic matter, quite diverse. So saprophytic fungi, some bacteria, they produce what we call humus, which is rich in very small molecules, but with a lot of organic matter. So bacteria are going to transform this humus and to allow the release of nutrients. And now the plants and the soil organisms can use it. And that's what we call a soil respiration. So it's a very simple image of what is happening with organic matter decomposition. And of course, in a system with a very intensive management, this organic matter source is very limited because farmers used to apply chemical inputs. So this is where, so they just put nutrients and they put massive doses of nutrients, too much, and they make the system totally unbalanced. And that's why we are getting problem, because soil diversity is going down. So as I said, soil structure is very important because by forming aggregates, it's a way to keep carbon in the soils. And this carbon can be used by soil organisms, and the aggregates, the production of aggregates can be facilitated by soil organisms, such as, for instance, of course, root plants, because of the release of some mucilage, but also, so it's like a glue. So this mucilage glues the particles, water, nutrients, and microspore, whatever. They can form aggregates. But also these aggregates can be stick to each other by fungal hyphae. And now we, at the end of the day, we start from the micro aggregates where we have microorganisms. We finish by micro aggregates, which are pretty big. And they are very important because it's a way to keep carbon safe. So, and it's very rich. It's a source of carbon. And that's why a lot of people consider that they try to promote no tillage practice, because when you do tillage, you break down this. So you release all the carbon stored in the macro aggregates, but also in the micro aggregates. So it means that the microorganisms living here are released and all the system becomes unsustainable. So that's why that's something very much important to understand when you talk about the carbon cycle. So just a very quick example of photosynthetic bacteria. There are not many, but they exist, and we need to mention them. Of course, we know about the cyanobacteria. I mean, in the paddy rice, you can see at the surface of the water, this green color. Usually they are cyanobacteria. So these bacteria, they can get what they need by using solar energy, CO2, atmospheric CO2, and water, like the plants, and they do photosynthesis. And they can survive in very trash conditions. And also they can form some symbiosis with other plants. By the way. So I'm done with the carbon cycle. I don't want to be too long. I'm just trying to show you the respiration. We sound academic, but after that, I'll show you some examples. So now let's go another very important cycle, the nitrogen cycle. So carbon and nitrogen. This carbon is nitrogen is very much important. Why is it important? Because nitrogen is a part of all organisms. And second, you see these sites for 6.20% of the dry.
Problems with the Dr. Didier connection, yeah? Isra?
Hello?
Yes.
Can you hear me?
Yeah.
Yes.
But your slide already closed. Can you
Or
right,
yeah, I don't
good.
know. I think maybe it's internet because I got disconnected
I
for
see.
1 second. Yeah.
I see.
Okay. So let me see if I can. So right now you cannot see my screen, huh?
Yes, but not yet. Sorry.
All right. Okay. Let me see. Because yeah, I don't know why I cannot see. Okay. Let me see. Let me try something. All right. So. Okay, so can you see my slide?
Not yet.
Not yet, sir.
Okay,
Oh.
just I think it should work now. Can you see?
Yeah.
Okay,
Yes.
so let
Perfect.
me put full screen. Sorry for that. Yeah. In Vietnam, internet is totally unpredictable. So
It's okay. It's okay.
okay, can you see my screen?
Yes, sir.
Yes.
Okay, perfect. So let me. I'm struggling with. Okay, good. So I don't know where did I stop. So I was mentioning about the importance. There is one. Okay, yeah. So about the importance of nitrogen, and so I explained, but because they are part of the amino acids, nucleic acids, and cell walls of bacteria. So that's why they are very important. They are involved in many mechanisms such as oxidation, reductions. So they are very key for mainly for bacteria, but also for archaea and some specialized fungi. So without nitrogen, life is not possible. So I'm not going to describe all the nitrogen cycles. I used to do that when I teach at the university, but for this webinar, I make it shorter. So I tried to on this slide to summarize what are the key steps of the nitrogen cycles. We start from the plants. So we have organic matter, as I explained you. So through the decomposition step, we have a release of nitrogen, mainly ammonium. And this ammonium is going to be transformed in nitrates and nitrate. Process is what we call nitrification or nitrifying. And these nitrates can be assimilated by the plants. Some plants are capable to assimilate nitrates, but also in some certain circumstances, anaerobic condition, for instance, this nitrate is transformed in atmospheric nitrogen, which means that the nitrate leaves the soils, and of course, it's a loss for the systems. On the other hand, when we work with legumes, for instance, they have the capacity to form some symbiotic symbiosis with rhizobia or other soil bacteria. So they can, in symbiosis, they can get atmospheric nitrogen. A part of this nitrogen goes to the plants. It's released also in the soils, and it goes to the ammonium pools and so on and so on. So this is how it works. And you can see for all the steps, microorganisms are involved. And for decomposer, they are soil organisms. So it's another way how to see the nitrogen cycle with nitrifications. We can happen with oxygen, but on the other hand, the denitrifications, which means transformation of nitrates to N2, this can happen only in absence of oxygens. So nitrogen fixation, I was talking about it. It used to happen mainly with legumes. We have some other species where have a capacity to fix in symbiosis with other soil microorganisms atmospheric nitrogen, and it makes this nitrogen available for the plants. So nitrogen fixation is a very important process with they catch this nitrogen from the atmosphere, where nitrogen is not limited. You have plenty of nitrogen in the atmosphere, so the plants can fix as much as they want. And of course, this nitrogen goes to the soil somehow, and it's very beneficial for the systems. And it gives also the capacity for the plants to grow in very poor soils. Even if they are nitrogen deprived, the plants can find by themselves the nitrogen she needs to grow up. And it's very much important. The fact is, you have two kinds of organisms involved in the biological nitrogen fixation. You have what we call the free-living microorganisms. They are able to fix atmospheric nitrogen by themselves. They don't need to form any symbiosis. I mean, for instance, a very good example is Azospirillum with sugarcane. Sugarcane used to grow in very humid area, and you have a lot of beneficial microorganisms, what we call azotobacters. And azotobacters can produce a lot of, can fix atmospheric nitrogen. And by fixing, a part of this nitrogen is released nearby the roots of the sugarcane. And it means that the sugarcane can benefit from this nitrogen. So it's a very beneficial association. It's not symbiosis, it's just association. On the other hand, we have this symbiotic association between legumes, but not only legumes, mainly legumes, with rhizobia, but not only rhizobia. And at the end of the day, what happens is that after some molecular mechanisms of recognitions between the two symbiotic partners, we have the formation of what we call root nodules. And inside these root nodules, the plants deals nutrients to the bacteria. And on the other hand, bacteria is capable to fix atmospheric nitrogens and release this nitrogen to the plants. So that's why we talk about beneficial symbiosis. Of course, this biological nitrogen fixation requires a lot of energy. And it's a strategy for the plants to fix atmospheric nitrogen. So basically, what does it mean? It means that if you put legumes in the soils, if your soils is very rich, full of nitrogen, the plants will give the priority to the normal assimilation of nitrogen to the roots, and she won't form any root nodules. On the other hand, if you grow your same plants in very poor soils, deprived of nitrogens, and you put rhizobia, the plants will start a strategy to form root symbiosis in order to get root nodules, where the rhizobia will help her to get nitrogen from the atmosphere, and they will make this nitrogen available for the plants. So that's why plants are very adaptable according to the situation where they are. So in terms of biological nitrogen fixation, I already mentioned some what we call free-living bacteria. So you have some Azotobacteriaceae, but also cyanobacteria. I mentioned that before because they are also capable to do photosynthesis. Of course, we have archaea, which are very much involved in this kind of mechanisms. And you have some example of photos showing this all these bacteria. But when we talk about biological nitrogen fixation, we automatically think about legumes and rhizobia. So as I explained you, it can be annual legumes, it can be tree legumes. 99% of the legumes can form symbiosis with rhizobia and other kind of microorganisms. And it's a way to get this nitrogen from the atmosphere. It's also why in agroecology, we promote the kind of intercropping between crops and legumes. And because in addition of providing nitrogen to the soils, legumes can also contribute in increasing the diversity of soil organisms. And I'm going to show you a good example of what we did in northern Vietnam. So this what is when you cut in two pieces root nodules. So the black part are the bacteroids, the bacteria where they fix atmospheric nitrogen. And it's dark because, as I said, this biological nitrogen fixation in the nodules can happen only if there is no oxygen. So the plant developed a strategy with what we call leg hemoglobin, which is something similar of the hemoglobin that we have in your blood. So this leg hemoglobin catches the oxygens and makes this area totally deprived in oxygen, and which means that the nitrogenase, the enzymes responsible for biological nitrogen fixation, can work, and this is how the plants can get the nitrogen, which is released here. So this is an example of root nodules, and when you grow the rhizobia, that's what they look like in Petri plates. So there are some other examples of plants. We can form symbiotic root nodules with bacteria, some parasponia. They are not legumes, but it has been demonstrated. And of course, if you go on the coast, I guess that you have this kind. We have this kind of actinorhizal plants, such as Casuarina in Indonesia. Indeed, very common along the coast. Plants coming from Australia, and Casuarina are capable, like legumes, to fix atmospheric nitrogen with actinorhizian plant with a microorganism we call Frankia. And you see an example of nodules with Frankia, and they are very much important. So if you are on the beach, you can look for nodules, and you will find some. So this is inside, you have Frankia, and Frankia is capable to fix atmospheric nitrogen in symbiosis with actinorhizal plants. Okay, I think I'm done with the academic parts. I don't know if, do you have any questions? I can see some comments on the chat. Okay, that's how to get the material. Okay, so I don't know if there is any questions.
Okay, thank you, Didier. So the participants here, 251. So if you are interested understanding more about what they're explaining by Dr. Didier, can yeah, go ahead. Is there any question to this? Hello?
Professor Burhanuddin Rashid, I think Dr. Junaid.
Yeah.
He raised hand, Professor Burhanuddin Rashid.
All right. Go ahead, Pak Bur, if you want to ask, please. Somebody,
Okay,
some of you are raised your hand, yes?
Sorry.
You don't need to allow us.
Okay. First, I want to test my voice. Is it clear, or you can hear my voice?
Yes, sure. Sure.
Okay.
No problem.
Thank you very much. Good morning, Dr. Reser and Dr. Junaid. I'm Rashid. I'm belonging of the Department of Soil Science, Faculty of Agriculture. Of course, I very interesting to this lecture, Dr. Reser, because this is very connected with my subject in my class. Actually, I'm conducting a class in soil quality and soil, water, and nutrient management. And this is a very important, I think, and very valuable lecture for us, as you explained about the function of the soil health in the relation to the soil agriculture sustainability. I have one let's maybe opinion about this this subject. I have some experience to let's see, teach our farmers about this issue. As you know that I think not only in Vietnam but in East Asia countries, they have some same problems about how to keep their soil degradation. In cases in South Sulawesi, I don't want to say about Indonesian situation, but I want to say about special in the South Sulawesi condition. We have very problem about soil health. What I want to say, there is one problem for us, that how to making a bridge between scientists and farmers. I have experience to teach our farmers on how to keep their soil health, but we have problem because farmers, they want to get high production, so they very serious to use chemical material to improve their production. And we have some research that this is very, very bad to our soil health now, especially in the soil in the South Sulawesi. What I want to say, based on your experience in many countries to keep their sustainability in soil health, if you have at least one word to develop the gap between scientists and the farmers on how to keep the soil health. Because I have problems to make our farmers understand on how the soil biodiversity or soil health very important to keep their sustainability, especially in agriculture environment. Yeah, maybe for this point, I want to maybe Dr. Reza Rafi, you have experience on make understanding between our scientists and the farmers. Sorry, that only one question. Thank you.
Thank you, Dr. Rashid. I mean, basically, we all face the same situation with farmers, and there are several ways how we can hope to move forward with these issues. I mean, but we all know that when you ask farmers to totally change the way how they have been growing their crops, it's not going to happen, because farmers consider that they know if they spend a certain amount of money, they know more or less that they will get at the end. And they don't have this vision about sustainability, because the condition, it means that their concern is tomorrow, is not the day after tomorrow, and so on. So let me give you some example about what we have done. Let's start by a very good example of something we did in Africa, because I was in Africa for 15 years before I moved to Southeast Asia. And in Africa, we have a lot of issues with soil health because soils are very poor. And if in Southeast Asia, you can get cheap chemical inputs everywhere, even in the middle of nowhere, in Africa, it's totally different. Chemical inputs are very rare and they are very expensive. So it means at the end of the day, farmers, they don't use them. And we were running a big project funded by Bill and Melinda Gates Foundation. It was in 3 African countries. It was in Kenya, Nigeria, and Ethiopia. And we were working on some alternatives instead of chemical inputs. They were what we call bioinoculants. It means commercial products containing beneficial microorganisms. And we put a lot of efforts and activities on bioinoculants for legumes with rhizobia, just from what I showed you a few minutes ago. So what we did, we started by demonstration trials. So we 3 countries where we were running projects, we set up in key provinces demonstration trials. So basically it means with the legume species that farmers used to grow, we set up trials where we inoculated some very good products. And from the trials, so the access to these demo trials were free of charge. I mean, free. Farmers can go, they could talk to the farmer association and whatever. And we organized field days. And during the field days, we were asking farmers, what do you think? What are you ready to do? And so on. And we were very positively impressed by the feedback we got from farmers, because they are the ones who came to us and asked, I would like to test this technology. I would like to test this technology. So we managed to organize farmer trials with farmer association. And instead of asking them, do that, they ask us, we want to do it. And it worked very well. And it's a way how we can contribute to adoption of this kind of microbial technology. Another example in Southeast Asia, in Vietnam, and I'm going to show you some results after that. It's we were asked to help farmers in northern Vietnam and slopes. People used to grow cassava, monocropping cassava intensively with a lot of chemical inputs. And of course, that the yields was going down gradually, and farmers were not happy because they were making less and less money. So, and there was room for cowpea, and because that's something they can easily sell on the market, and the price is pretty high. So we started intercropping, just intercropping. We didn't change anything. We started intercropping cassava with cowpea, and we set up this kind of intercropping trials in several places. I don't remember, in many farms in one commune. And we organized field days with local authorities and everything. And we asked farmers, what do you think about this intercropping? I mean, not 100% of them, of course, because it never happens. But the big majority of these people, they said, we were very happy with that. So it means after 1 year, the second year, I don't remember, but we increased by maybe, 50%, the number of farmers asking for using legumes. And because they consider that it was good for their soils, I'm going to show you the kind of results we got. And the next step will consist to say, okay, guys, you have legumes, which is good, but stop using all your chemical inputs and try to see how you can benefit more from the legumes. Because for them, you put the legumes, you get money from the cowpea, but they keep applying their chemical inputs and so on. So it's a gradual process, but there are 2 success stories I can share with you. And I was in Central Island in Vietnam maybe 3 weeks ago, where they have big issue with coffee. They have been running coffee production for 25 years and intensively. And now they have to replant coffee plantations because the trees are too old and they cannot. Why? Because of this intensive management, soils are full of nematodes, bad nematodes, Meleidogene and Pratylancus. So all the new seedlings they plant, they died because of the nematodes. So when we talk to farmers, we told them, okay, guys, there are many options how we can do it. But they say, oh, I don't want to change. So I said, okay, you don't want to change, but what is going to happen to you? But most of them, what they do, instead of trying to sort out the problem, they shift to another crop and they start growing fruit trees, like they did with black pepper. But the soils remain the same. And after a few years, they will get problem with soil-borne pests and diseases for sure. So we try to work closely with them through this big project funded by Australia, and to convince them there is time for changing. And if you don't make the changes, you will be in trouble. But it's a long process. And I think this gap you pointed out, we are gradually trying to fill it up. And because there is, in some circumstances, there is no way farmers have to change. Somehow they have to change. I'm going to show you at the end of my presentation, because usually we think that organic management is good for the soils, but is not good for the farmers because they lose money. I'm going to show you what we are doing right now on green tea in northern Vietnam. And you will see that at the end of the day, farmers growing organic tea make more money than farmers doing conventional tea. And you will see the data. But I don't want to talk too much because it's getting late and I don't want to delay the schedule. I don't know if I answer to your questions, Dr. Rashid.
Yeah, it's okay. So it's a very nice experience because this is very valuable for me because now I'm working for the some community in some areas, and now we are trying to arise one issue now that we call zero waste by production. So we are going to use all parts of the plants. And especially, in Indonesia, we have problem in some plantation, a big plantation, yeah, especially in the, oil palm plantation, and they have many waste there. And now we are going for the manage. And when we are, as you say, we are now try to make inoculant and manage their byproduct, and we are used as a compost, as biochar there to improve our soil quality now. Okay, thank you very much, Dr. Lesher. Please, but Junaid, could I want to one question more? I think
just drop because of many people want to ask and give a comment into the slide also. Okay. So can you drop just into the chat room, please?
Okay.
Okay. Dr. Didier.
Yes.
So we have plenty of questionings.
I can see that. Yes. Yeah,
okay.
I can see.
So because your slides are very interesting, right? And these questions and comment from Kifayanti. Her question is regarding virus in the soil, in the interaction with other microorganisms in the soil, including mycorrhiza. Does it mean that effectivity of fungi cannot be lower when apply the virus infected soil? How can we detect if the soil has some virus? Is it clear?
Yeah, it is. It
All
is.
Right.
I mean, what kind of relationship between mycorrhiza and P availability in the soils? We'll try to find some other strategies, and forming a root mycorrhization is one of them, because mycorrhization enhances the P nutrition by increasing the surface of exchange, where we will be able to get phosphorus from the soils. They can also contribute to make more phosphorus available, because we all know that there is a lot of phosphorus in the soils, but a little part of this phosphorus is available for the plants. So there are several ways how we can make this phosphorus available for the plants. So far, what we have found is because we have been working on mycorrhiza. I didn't present slides on mycorrhiza because it was just 1 hour, 30 minutes. It was too short for me to go through all the things. But if we take the example of rubber trees, which is important in Indonesia, we have run investigation in Thailand, Northwest Cambodia, Southern China. We are planning to do it in Vietnam, and I hope that we will be able to do it in Indonesia. Rubber trees, they used to form a lot of root symbiosis with mycorrhiza. I mean, in sandy soils in Thailand, in Northeast Thailand, the mycorrhization of the roots was about 70%, which is pretty high. So I mean that rubber trees, usually there are some controversies, but people consider that there is no need to fertilize the trees. We do it for all pine, that Dr. Rashid was talking about. And of course, the alternative of using, I visited many companies in Indonesia, from Medan to PT Smart and so on, where they were doing their own compost, and instead of limiting the utilization of chemical inputs. For rubber trees, it seems to be more basic. Sometimes people apply a little bit of fertilizer, but it means that the plant doesn't have a choice if she wants to grow up. So she tried to make a lot of, take advantage of a native mycorrhiza. And that's why we have about 70% of the trees which are naturally, are formed naturally symbiosis with mycorrhiza. But mycorrhiza alone, it seems not to be a very relevant indicator of soil health, because it used to happen, whatever is the soil health condition, is more dependent of the availability of P. So basically it means mycorrhization can be very limited in soils where farmers apply a lot of P fertilizer. So it's not really related to soil health. So I can see other questions.
Are we going to the next question, Didier?
Yes, please go ahead.
Okay, so this question, as we know, all agriculture has serious problem with soil-borne disease. The question is simple, but maybe the answer is difficult. How can we ensure that the soil biodiversity under our plants are those beneficial and not vice versa? For academic scientists like us, we can simply take soil samples and test them in our lab. But for small farmers, smaller farmers in the farmers level, accessing lab or scientists for them might not be possible. So what do you think about this question?
I mean, it's very important societal questions. Everyone is asking for soil health indicators, but farmers, private companies could quickly measure to have an idea about the soil health of their plantations. It's a very simple concept, but not so easy to make it effective and successful. And I mean, right now, we need more investigations to identify some key indicators allowing farmers to have an idea about what is happening in their soils. But basically, I mean, usually where soil diversity is pretty high, yield is pretty good. And you have a lot of diversity, you can see by yourself. I mean, when I was working on this project with cassava, everywhere we sampled with monocropping of cassava with plenty of chemical inputs, when we looked for soil fauna, we didn't find many. On the other hand, in farms where we had this intercropping with cowpea and cassava, it was so easy, even with the same rate of chemical inputs, it was so easy for us to find soil fauna. So, I mean, it's kind of very simple observation that farmers can make, and it can help them.
Yeah.
Even if the system is not optimized, it's easy. Yeah.
Right. Well, so the next question is it possible soil microbes increase soil quality with many inputs of pesticides? How long estimate time needed for the process? The question from the Ibu Sri. Yeah.
Yeah, I mean, so yeah, it depends. It depends. I mean, it depends on the situation. If your soil is highly polluted with pesticides, it may take some time. But as I told you, I'm going to show you the results later. You will see, second part of the presentation. I mean, just introduction of one legume species, cowpea, has a significant impact on the soil diversity for microbial communities and soil fauna. So it means even when people were, when we went in the field, we saw many places where we saw the granules of chemical inputs. So I mean, basically, if your system is becoming more sustainable, even if in the past they use pesticides, you may get an impact quite quickly. But it depends from one place to another one, of course. I cannot say I'm sure at 100%.
Oh, well, so the next comment and questions is, okay, so some response, the question is very interesting, then thanks to your explanation, it's very interesting. And still a question from him. I have about the condition of microbe between dry land and wetland. So please give us opinion about these those lands.
Yeah. I don't know what does it mean. Does it mean that for the same place, what is happening in terms of soil diversity during drying condition and wet condition?
I think so.
Sorry, Doctor. Sorry, Pak Junaid. This is my question.
Yes.
Yes. Maybe Dr. Lesher, we have an expert about the swamp area.
Yeah.
Yeah. You know, in Indonesia, we have a special plant called sagu plant. Maybe you know about that?
No, I don't know.
Okay. Actually, this plant is living in the the lowland, but now we have going to make a focus of these plants because yes, now is one of the food resource for us for the next for the future maybe. So now we want to try to let's see to more study about the microbes around this environment. So maybe you have some, you can teach us some, let's see, how if you can say the composition of some classification of the microbes around this environment, between differences between the dry land and let's say for wetland. It's not in the paddy field, but this is a special area in the swamp area, and some special plant grow there.
So you mean you are talking about peatlands?
Yeah, it looks like peatland.
Okay. Yeah, because yeah. So I see two questions in your question, by the way. The first one is which kind of approaches we can use if we want to investigate the microbiome of this new plant you are talking about. So this one is there is no big issues because we by using when we want to assess the microbiome, we do what we call Illumina sequencing, for instance, and we run these kind of things. We work with private companies and we do the bioinformatic works, and we can come up with a map of all the microorganisms contained in your soils, and you compare with or without your plant. So there is no problem. You may get problem with DNA extraction because peatlands are very specific and soil characteristic may be a problem, but there are some adjustment to do. Now, regarding the fact that your peatland system is very specific because there is a lot of, mainly it's anaerobic conditions and everything is stabilized. That's why you have a lot of carbon in the peatlands. When you start drying this, you change everything because you make oxygen available. Once you make oxygen available, it means some soil respiration, which was not possible before because it was anaerobic, becomes aerobic. And by doing this, that's why you are going to get mineralization of your organic matter. So by getting this on, automatically you are going to see new microbial communities coming up because the situation will be different. So that's why I was asking you, because I attended the webinar on peatlands some weeks ago, and that was one of the issues. What are the main consequences when people start drying peatlands? And in terms of soil microbiology, I mean, the main consequences for me, and maybe I'm wrong, but for me, it's the fact that you change the condition. We move from anaerobic condition to aerobic condition, and automatically you are going to make changes on the microbial communities with consequences on the carbon sequestration, because automatically this carbon we will store in anaerobic condition is going to be consumed. So there is no more storage of carbon and so on, but carbon is going to be released.
Well, Didier,
Yeah.
still many questions here. So can I go to the next question, or do you want to show the next slide?
I think
Thank you very much.
it's up to you.
All right.
I can maybe continue, and if we can finish by all the questions to be sure
Okay.
that we will have time for all the slides.
Right. One more question, and then go to the next session.
Okay. Sounds good.
The question from Irshadi. So he is a doctor student in Unhas, environmental technology. How the process of rock weathering affect for organic matter, soil fertility, and variety of vegetation that goes on the soil? Can you?
Oh yeah. So when
Okay.
when I teach at the university in Hanoi, we have 2-3 hours courses. So one is mainly on the soil diversity. So basically a part of what I showed you this morning, and the nitrogen cycle and carbon cycles. The second one is about oxygen cycle, phosphorus cycles, and for the oxygen cycles, this is something very much important about rock watering, and also for the phosphorus, because phosphorus cycle, because phosphorus comes from this rock weathering. But I mean, it's a bit tricky to answer to this question because it's a very long process. It's not something we used to happen very quickly. So to be honest, how the process of rock weathering affect the organic matter, soil fertility and variety of vegetation, it's I don't know, because I think from my understanding, it's you can make some conclusion only if you work on the long-term trials where you follow up this kind of parameters on the long term, and I don't have any knowledge on that. I'm sorry. I'm not going to say stupid things. It won't be relevant.
Still several questions, but we give a chance to show your the next slide if you have, Didier.
Okay.
Please go ahead.
Okay, sounds good. So after all this introduction, a bit academic introduction, so there are words, there are academic knowledge, but what does it mean? So now we are part of this Asia Pacific CMBP network, and I would like to explain you what is CMBP, because of course, Dr. Junaid, you know, because you are the one who made this possible at your university. But I think for all of you, and I'm quite impressed by the number of people we have right now, 245, very impressive. So CMBP is a network, and this network aims to gather people sharing the same interest on agroecological practices and microbial technologies for enhancing soil health and making agriculture sustainable. So we have this website where we try to gather information we can share to all the members. And so you can see that's the page today, just about this Top Scientist webinar series. And we used to put briefs, but you have also here information about the network, objectives, list of partners, contact of partners, and everything. So we try to gather information that we share between all the members. So we have about 60 members coming from 18 different countries in Southeast Asia and beyond. And why are we together? So let's go back to the beginning. When I arrived in Hanoi, I was asked to set up a microbiology lab. When I asked, where do you want this microbiology lab set up? It was this room, you can see here, but it was a paradise for dust, garbage, and everything. It was not used, by the way. So we decided to set up a microbiology lab, but a real lab with benches, with equipment. You can see this, on this slide, this is where we do molecular biology. This one is where we do microbiology. And this is where we have on this bench, we do everything about microscopy, when we score the root staining from mycorrhiza and everything. So we started by setting up a common lab, share with CIRAD, CIAT, and AGI. That was in February 2018. It was a very good starting point with official launching and everything. But 1 year later, we organized a meeting in Hanoi where we invited partners from Vietnam, of course, but also China, Thailand, Cambodia, Australia, and France. And we all agreed about the relevance to create a network instead of just a lab. And that's where CMBP was born. And it was quite good because we were all willing to promote soil microbiology, soil health, and agroecology. So this is where we were 1 year later. So you can see you are here. Where is Indonesia here? You are here. We have more partners from Indonesia, but I mean, this situation is completely changed because we have more members who join us now. And of course, if you want to get an update, I invite you to go to the website and to check what are the members. And you can see most of them, of course, because that's the target, comes from Asia, Southeast Asia. We have some private companies also, which is pretty good to get the private sector on board because they have some practical concerns. We are happy to work with them to sort the problem out. And it's easy to get in. It's easy to get out the network if we are not happy. It's free of charge, but at least what we want is to gather a consortium of people sharing the same interest, and we have to see how we can work together. So it doesn't mean that it's supposed to be Dr. Jeanette working with Didier, Dr. Rashid working with Didier, but maybe Dr. Jeanette working with Dr. Long in Vietnam, or Dr. Rashid working with Dr. Gift in Thailand. So that's also the kind of things we want to promote, because in all these countries, people used to share the same concerns about soil health and everything. So that's the objective of the network. Okay. So as I said, main mandate is promoting agroecological practices to enhance soil health, because I think we know that if farmers keep using chemical inputs intensively, they will go straight in the wall, and we want to do something before it's too late. I was mentioning this example of coffee sector in Central Island in Vietnam. I think they have crossed the threshold, and I hope it's not too late, but I can tell you that to come back to a better situation is going to be time consuming, and I'm not sure we will be successful, especially if farmers are not very happy to change their minds. But anyway, so we want to anticipate, and we cut across the commodities. We have ongoing project on coffee, tea, rice, pepper, soybean, cowpea, cassava, rubber trees, not yet on banana and sugarcane, but maybe in the maybe on oil palm. I really tried on palm oil, and with private companies, it's not very easy because they believe they can do everything by their own. But anyway, we keep trying, and next week I'm going to Mekong Delta in Vietnam to work on coconut plantation. So because there is need also. So if I try to summarize a bit of what we are trying to do through CMBP, promoting agroecological practices, So it may be intercropping with legumes. It may be utilization of cover crops. It may be also utilization of bioinoculants. So that's a kind of what we call microbial biotechnology. We can use what is available in terms of commercial products, but also in some cases, we can work on the selection of a beneficial microorganisms, and we can also formulate the bioinoculants when it's necessary. If it can be for legumes with rhizobia, but also in terms of biological controls to eliminate soil-borne pests and diseases. We put a lot of effort in capacity building. Right now, we have 2 Vietnamese PhD students. We have 2 undergraduate Vietnamese students. Thursday, we are going to get interview for a new Vietnamese PhD student. We have very good partnership with Deakin University. And we do in-country PhD. So I mean, the PhD students are based full-time in Vietnam, but they are registered in Deakin University. And during their PhD thesis, they can go to Melbourne for 3 to 6 months, fully paid by Deakin to learn techniques but most of the time they are in their countries. So it means they can dedicate themselves on field activities. They can work on the local situations, which is not possible if you do your PhD in Australia, for instance. I saw a presentation 2 years ago of a Vietnamese who was working on soil-borne pests and diseases of coffee, but he was in Sydney, so he had to work on soil-borne pests and diseases in Australia, but was not really relevant for the situation here in Vietnam. So that's why this kind of package is very successful from our opinion, by the way. And we tried also to promote soil microbiology in existing curricula, or if we are requests, we can also contribute to create new curricula for promoting soil microbiology to undergraduate students since the beginning. And we are trying to work on to come in terms of support for private companies when they have questions about soil health indicators. And we have this lab. So I show you some pictures, and you can see the kind of activities we can run. Everything is available on the website. I'm not going to spend too much time on this. So now I'm going to finish my presentation about 2 case studies we run in Vietnam. So the first one is about intercropping legumes with cassava in northern Vietnam in Giang Bai province. And the work was done by this gentleman is Chung. He's lecturer at the Vietnam National University of Agriculture in Hanoi, but he was registered in Kasetsart University. So he did his PhD with us, and we work in this commune. This lady, a fantastic lady, she's the president of the farmers, and she was very helpful. So we work with them. So no research station, we were working in farms directly. And it was very nice to interact with farmers. So as I said, a lot of issues with monocropping of cassava in terms of soil erosion, in terms of utilization of massive amounts of chemical inputs. So in Giang Bai province, and this is what we did. You can see the kind of landscape. You can see it's not too steep, but it's quite steep. And when we prepare the field, so the soil is totally naked, no protection. So that's why when it starts raining, you imagine the consequences of soil erosion. So here we tested the intercropping between cassava and cowpea. The farmers are the one who chose. They decided to use cowpea. It's not us who were suggesting cowpea. And we identified 9 farms doing monocropping, 9 farms doing intercropping. And we assessed the soil fauna and the diversity of the bacterial and fungal communities by Illumina sequencing. So in terms of results, you can see in terms of microfauna abundance, the huge significant difference between intercropping farms and monocropping farms. In terms of abundance, highly significant. In terms of microfauna, we also got a very positive impact in terms of richness, in terms of diversity, in terms of evenness of the intercropping compared to monocropping. For bacteria, we have better richness and a significant change, positive changes in the structure. But on the other hand, no impact on fungal communities. So once again, we didn't ask farmers to change anything. They did their usual management. We just introduced the legumes, and this is what we got. So it gives you some ideas. If you reduce the utilization of chemical inputs, you promote the biological nitrogen fixation, because we didn't apply any rhizobial inoculants. We had some rhizobia, native rhizobia, but there is room for improvement. But in terms of soil health, through soil, fauna, and bacterial communities, we have got a huge impact of this intercropping. The second one is still a PhD thesis made by this gentleman, San Leviette. Is also registered at Deakin University. The idea was to compare organic tea with conventional tea in Thanh Nguyen province in northern Vietnam, which is the key province for green tea in Vietnam. So you can see the impact of the two kind of practices, and of course, how it affects soil health. So if you are interested, we have published some review papers, one in 2021 and one in this year. And the research question was to see how does this kind of management used to impact soil health, of course, but also tea productivity and tea quality. So we identified in one commune, very well known for quality of the tea, plantations with organic management and conventional management. And you have for each, you have what we call conventional, we have what we call converted and non-converted, because people started growing tea and they were growing rice, but somehow they noticed that they were making more money with tea than with rice. So they stopped growing rice. They brought soils from the hills, and that's what they call converted field. And you can see that here, a lot of, we work with a cooperative where farmers grow organic tea and they apply a lot of organic matter, as here. On the other hand, here, utilization of chemical inputs and everything. And what I was telling you about what we call converted and non-converted, but you can see the huge surfaces of lands dedicated to tea. So what is happening when we compare the two kind of practices? We have a significant impact on the abundance, richness, and Shannon index of soil macrofauna, but also because we assessed the mesofauna using two protocols, one with centrifuge extraction and one with the classical one with funnel extraction. But I mean, in terms of abundance for both, richness, almost for both, yeah, it is significant. And it is also significant for Shannon index. We have a huge positive impact of the organic management compared to the conventional management, which is very good in terms of sustainability. So you have more details about the abundance of the soil macrofauna and the abundance of the soil fauna, the richness, Shannon index, and you can see it's always higher with agroecological practices compared to conventional one. So as I was telling you, and I think it will be my last slide, is what does it mean for farmers? Because it's nice if you are able to protect your soils by enhancing the soil health, but if at the end of the month, at the end of the year, he gets less money, it's not going to work. But you can see here, this work has been done in 2019, 2020, 2021, and this is the main. Every time, by doing agroecological practices, farmer was making more money with conventional practice. So I mean, from the idea where we say organic management is good, but it's a way to lose money. We can see that at least for tea, but we hope that we will do this kind of similar studies with other commodities. With tea, in where we work, that's not the case at all. And I think that's also one of the message we can send to farmers. When there is market, organic products, you can sell them at a higher price, and you use less money with chemical inputs, and you get more benefits. That's the kind of message. I know it's not so simple, but yeah. Okay, I think I'm done with slides.
Thank you very much, Dr. Didi. We still 3 questions remaining. So, yeah. So the question from Andi Linga. So from Soil Animal Sciences, what your opinion about improving soil health with grazing lands? I think grazing, yes, these animals. Yeah.
I mean, I don't believe that the action of grazing is improving soil health. But on the other hand, if you have animals and if you want to feed your animals, maybe you can include some, I don't know, what do you mean by grazing? Is it rangeland grazing, or it means that you use some biomass to feed your livestock?
I think so. Yeah, grazing means food for animals, I think, yeah. So yeah.
So it means in this case, because do you have some pasturage in Indonesia?
We have in Hasanuddin University, we have a center of excellence for pasture. And so not just only growing animals, but also processing post-harvest management there.
Oh,
so,
okay.
Yeah. Someday, if you are visiting in Unhas, we will see the grazing land in Unhas.
Yeah, I hope that now, because things are getting better with COVID, it will be much easier to travel. Yeah, definitely. Yeah, I mean, but if you incorporate, I mean, if you take into consideration the grazing, livestock, and everything in your system, of course, it will positively impact soil health, because you will introduce some organic matter through the animal feces, and it will automatically impact positively your soil health, because you will add organic matter to your soils. And even if you do a kind of recycling of what is all the waste coming from your livestock, and you introduce them to your field, it will be beneficial. Yes, definitely, yes.
Right. Yeah. So next question is how much profit is obtained from the legume, example soybean, compared to the nitrogen fixation microorganism obtaining water and nutrients from the legume?
I think what is important is be sure that there is market. If you decide to introduce a legume in your system, you need to be sure that there is market for the final products. Because if you just grow, it depends what you expect. I mean, in the case I gave you, people were harvesting, the grains of cowpea, and they were selling them on the local market. But instead of getting a grain legumes, just to go back to your previous question, you can grow forage legumes. A lot of farmers grow this kind of forage legumes in northwest Vietnam, in the mountains, because they have livestock. And you prune your forage legumes, and you can feed your animals with it. So you take advantage of the final products. So the question we had was we were very successful with cassava producers. We were not successful with maize producers because in addition of utilization of water and everything this person was listing, there is space issues. And farmers growing maize, they didn't want to change the way how they were growing maize. We consider that, let's say, in 1 square meter, they have 2-3 lines of maize. And if you include the legumes, you need to skip at least 1 row where you will grow your legume instead of maize, and they refused. So what we did is we keep the same layout, and in between the very teeny rows, they planted the legumes. Of course, it was not successful because it was too tight, and because of the competition with the light, you imagine, with maize, compare maize with cowpea and everything. After a few weeks, the cowpea was gone. But I mean, it's complex systems, but you need to be sure that farmers will benefit from whatever they introduce in the new system of production.
Well, so is there any soil cultivation technique to support development of beneficial soil microbes?
Oh, yes, I can see. Yeah. Yeah, I mean, you know, people were struggling with phytopathogen nematodes, such as melaidogene, for instance. A best way, instead of applying chemical nematicides, the best way is to apply organic matter. You apply organic matter, automatically you will impact on the soil health, because usually nematodes, they take advantage of free space. When the soil biodiversity gradually goes down, there is a lot of room for other organisms. And nematodes are very opportunistic. So when they see free room, they get in, multiply themselves, and that's where they become very, very difficult, very tough soil-borne pests and diseases. But on the other hand, when you apply organic matter, you start to create new soil diversity. There is less room for nematodes. There is competition between beneficial microorganisms and nematodes, and it's a way how you can improve gradually soil health. You can also raise the pH. Dr. Rashid was talking about biochar. We are working on biochar as well, trying to increase soil pH. So by increasing soil pH, you make soil conditions more favorable for soil biodiversity. Yes, there are several ways. And of course, you can also apply commercial bioinoculants with effective microorganisms. Yeah, that's also a way.
Okay, so last but not least, this is my comment and question. So, so you show us your lab, yeah, your collaborative lab in Vietnam is very interesting, clean, and if looking at here, so very complex tools. So, do you think that is it possible us to join or at least working with your lab and then understanding about what the biomicrobiomes in our region, for example?
Yeah, of course, of course. I mean, I can give you some examples. One of our Thai partners from Chiang Mai University, she was doing a PhD, and she was working on the microbiome of rice, comparing organic versus conventional. So she asked to come to our lab for 3 months. So she came with own soil samples. And she came also with some funding, because we can take care of the normal basis of the lab, but we cannot pay for your analysis and investigation and everything. All the equipments are available for who need to use them. So she came with her own budget. We did the DNA extraction with her. She was trained. She worked on the bioinformatics analysis.
Right.
And right now she's finalizing the publications. So, I mean, this platform is also to give opportunities when you have some funding, opportunities to some of our members to come to get training. So in this case, it's my colleague Laetitia, who is in charge to work closely with our members. But of course, you are most welcome. And we can also, when it's possible and when we can also co-supervise some students when English is not an issue, because that's the main problem we have in Vietnam. But you can see we have already 2 PhD students, and we are hoping to get a 3rd one. So because with a good English, because it's required, as you know, because you did your PhD in Australia, the requirement in English is pretty high. So of course, there are several ways how you can take advantage of this technical platform in Hanoi. Now the borders are open. They have been closed for 2.5 years, but not anymore since March 15. So yeah.
Thank you very much. So I think this is a good news. And then we can move closer with Unhas and then your lab. So the second question is, I think that's some question asking that your slide should be, is it possible to share your slide to our audience here?
I can share almost 90% of the slides. The ones I won't be able to share with you is the last one about the
to the ISRA, the international office.
Yeah.
You can manage your slide. What do you want to share? What do you will not to share? No problem.
Yeah, I will.
So actually, all your slide is very interesting, very inspiring, so very unique. That's because you combine with the video, your photo, and then so easy to follow, easy to understand. And thanks for that. And I don't want to conclude this the slide because it's very clear. But in my opinion, that's what do you want? What you say that there are a nitrogen cycle, carbon cycle, and what the role of the microbes in the soil. So that's why if we maintain the microbes in the soil, so we can get a healthy soil and support to the sustainable agriculture. So this is my third comment. And also my students also undertake the soil microbiomes and understanding what the difference between the different biodiversity in land with disease, plant disease, and organic plants. So very clear, I can see the data is a very huge obtained microorganism in the organic soil, and very weak, very weak, less microorganism in obtained from the plant disease. So in the also we have a student undertake also the research about the biochart. We can see the indicator biochart that we have a biochart, so less infected plant. That's a good consequence. And then I think I agree with your slide, and that's we just saw evidence that if a lot of microbiomes, diversity populations and species, they can drive for healthy soil, healthy plants, and healthy environment. So this is my last comment, and thank you. Thanks once again, Dr. Didi. So I will be back to Isra. Isra, do you want to say something? Dean of Agriculture still here. Maybe they want to close something. Yeah.
Let me.
All right. If our Dean of Agriculture Faculty, if you want to say something before we continue to the other session. Professor Dr. Salenki. All right. All right, and now we should move to the our agenda. We want to say thank you very much for Professor Dr. Didi Lesser and and give you and now we want to show you the certificate appreciation for our Vice Director of Research, Innovation and Partnership, Universitas Hasanuddin. Well, I want to share my screen. Here, thank you very much for your valuable knowledge to our participants. And then I will send this certificate to in your email soon after this webinar. Thank you very much, Professor Dr. Didier. All right. Now, then let's take a photo together. So please, all of the participants, open your camera so your handsome and beautiful face. All right, I want to count 10. I think most of you have opened your camera. So I want to count. All right, now we have 12 pages. So, well, all right. I have to say 1, 2, 3. Cheese. Second slide. 1, 2, 3. Cheese. The third. 1, 2, 3. The fourth, 1, 2, 3. Yeah, we have lots of participants here. The fifth slide, 1, 2, 3. 6, 1, 2, 3. Seventh, 1, 2, 3. 8, 1, 2, 3. The last one, 1, 2, 3. All right, so we have done, have taken photos together. And now finally, from the deepest of my heart, we do apologize for all mistakes during this event. Thank you very much, Dr. Dede, for your kind attention. And thank you very much for all of the participants to spend your time in our valuable webinar today. It's a lot of participants here. I hope, and we hope, Universitas Hasanuddin, all of you can get more information and knowledge about our speakers' materials and webinar today. So at the end of the session, how to close the webinar. Thank you very much, Dr. Didier. I will to send you the certificate soon and the photos of the webinar today.
Yeah, thank you so much. I want to say thank you very much for this nice opportunity for sharing my concern about soil health with so many people. So it was very nice. I do hope that it's the beginning of a long series of collaborative activities. I've been talking with Dr. Jeanette for some time about that. So you have a better idea about what we have been doing. So the door is open for other collaborative actions. I mean, we can think about if you have some needs for teaching on some specific topic related to soil health, soil microbiology, don't hesitate to let me know. This is something quite easy to organize. We are talking about supportive projects. So I think that Sulawesi is a very specific place in Indonesia where you have a lot of needs on the soil health. So let's try to figure out something. It can be through students or collaborative project. I mean, I'm trying to initiate things. Now the ball is on your side, and don't hesitate to let me know when you need something from me. So it will be my pleasure, of course. Thanks again. I really enjoyed the webinar. Being with you was
Thanks
great.
once again, Dr. Dede. I do appreciate what you offer. Yeah. So actually, very close. We will again organize international conference. But if you I mean, if you still become our main resources of our international conference in upcoming year, so we are really happy and then we appreciate with that. Because of, so now over 250 participants and they enjoy with your slide. I think there's so many, a lot of questions not answered yet. But if we're doing more detail with the conference, for example, we are really happy. So thanks once again, Dr. Didi. Yeah. So when you are coming to Makassar?
I don't know, but let's hope. Let's hope this year.
All right. Okay,
be my
so
pleasure.
keep in touch.
Yeah,
so
definitely.
Keep in touch, and then Isra will also send something like that. All right. Well.
Okay. Thank you so much. Have a good day.
No problem. Yeah. Bye bye.
And all the best for the Ramadan.
Thank you very much.
Bye bye.
Bye bye. See you.
Prancis.
Isra, jadi nanti anunya ya, kita kontak aja Didir untuk slide-nya. Jadi mungkin dia seleksi slide mana yang mau diberikan ke audiens. Jadi nanti Isra mungkin di Google apa gitu ya, di Google Drive kalau supaya bisa diakses. So make it easy.
Iya Pak, nanti rencananya mau di Google Drive semua recording filenya sama file apa